Hello again, people. What we have to talk about in this video is serial dilution. And that is a thing that happens in microbiology. That is a major tool in microbiology that you don't need to do in very many other fields. So let's get into serial dilution. I'm going to set up a hypothetical situation. We are going to do some analysis using some random made-up hypothetical analysis machine. And it only works if we put in a sample that has between 30 cells per milliliter, 30 live cells per milliliter, and 300 live cells per milliliter. Okay, so what we are trying to do is generate a sample of bacteria where we have a solution with 30 to 300 cells per milliliter. Somewhere in that number, it's going to be okay. If it's not enough, it won't work. If it's too many, it won't work. So it has to be between these two numbers. This is a thing that happens in a microbiology lab. Literally, these are the numbers we use as targets a lot of the time. So, okay, so how do we get that? And why is it hard? Why are we talking about this? We're talking about this because if you have a cloudy growth medium, if you have a cloudy culture, I told you that if there's turbidity, it's over 10 to the 6th cells per milliliter. But you might also know that if you let a culture grow overnight, it might be more like 10 to the 9th cells per milliliter. And this might be a 10 milliliter culture. So you might have 10 to the 10th cells. Who knows? Some of them might be dead. They might have grown up all night and then half of them died, or like 90% of them died. So you really can't figure out how many live cells you have very easily. So what we do is a serial dilution. And let's say further that I know how many bacteria are in this, because I'm making this up, but you are doing the analysis and you don't know. So let's say the real number of live cells in here is 5 times 10 to the 8th cells per milliliter. So that's the real number, but you don't know that. What you know is you need to somehow dilute some unknown population until you have a liquid suspension that has between 30 and 300 cells per milliliter. Okay, so how are you going to do that? Well, that is where we do serial dilution. Serial dilution. And what this is going to do is let us generate samples across many orders of magnitude, and one of them will have the right number. So what we do is we set up test tubes that have 9 milliliters of broth or water in them. So let's say we set up 3 of these. So 9 milliliters there, 9 milliliters there. And these are sterile. And they have little caps on them to keep dust out. Okay, so what do? We take 1 milliliter of this unknown suspension and we add it to the 9 milliliters. So our final volume here is the 9 milliliters we had plus 1 milliliter of our culture. This is a 10-fold dilution because we went from 1 milliliter of our culture to a total of 10 milliliters of liquid with our 1 milliliter spread out in it and diluted in it. So since I knew the original concentration of cells, or the population size, we would say, I know that this is a tenfold dilution. So this is 5 times 10 to the 7th cells per milliliter. You don't know that. You just know you did a tenfold dilution. Well, this is a serial dilution. So once you've mixed this up nicely, and it's achieved whatever the number it is, you're going to take some of this. You're going to take 1 milliliter of this and add it to this one. So you're making a tenfold dilution of this one. And so once you mix this up and shake this up, you have 5 times 10 to the 6th cells per milliliter. So once you've really mixed this up, you can then take some of this and put it in this one. So, sorry, I've been skipping writing plus 1 mil, plus 1 milliliter. And again, this 1 milliliter gets diluted to a total of 10 milliliters. So that's a tenfold dilution. 5 times 10 to the 5th cells per milliliter. Now, a skill we get when we do microbiology for a long time is figuring out what dilutions we have to do. And so we know this is not enough. We know we have to keep going. And I'm going to just, I'm just going to draw these. I won't put as much detail into them. But we keep going. Do another serial dilution. To get 5 times 10 to the 4th cells per milliliter, and then put this one in there. Sorry, 1 milliliter in there, and now it's 5 times 10 to the 3rd cells per milliliter. This one goes in there. This one is 5 times 10 to the 2 cells per milliliter. And we do one more, because we aren't sure. And this is 5 times 10 to the 1 cells per milliliter. So what we're going to do is we're going to analyze with our analysis machine up here. We're going to analyze several of these tubes, and whichever one had the number in the target, that one's going to give us good data, and we'll see it. So we need one of the tubes that has between 30 and 300 live cells per milliliter. So if we put that in scientific notation, this gets a little easier. So we need between 30, which is 3 times 10 to the 1, and 300, which is 3 times 10 to the 2. That's what we need. So have any of these tubes reached those concentrations? Let's see. What's this one? 5 times 10 to the 2, that's 500. That's too much. But this one is 5 times 10 to the 1. This is 50. So this is between these two. So this one actually has the right concentration of cells. So if I were doing this and I then put this in the analysis machine and I saw this had the right number of cells, so it's able to analyze them correctly and give me the right data, I would be a little bit embarrassed that this was the right one and all these other ones were wrong. Generally what I want is to have the correct concentration be one of the ones in the middle, so I would have wanted to keep doing dilutions. But it doesn't matter because we found one at the right concentration. So another thing you might ask is, why didn't we just do a millionfold dilution? And there are a lot of reasons for that, but one of them is that we can't measure tiny volumes we would need to do a millionfold dilution. We can't do that with a lot of precision. And also, we wouldn't know, should it be a millionfold, or 10 millionfold, or a billionfold? We just wouldn't know. So this is the process we have of making many different dilutions, and we can then test each one to see which of them has the right concentration of cells. And one of them will. That's the beauty of it. One of them will have the right number of cells. So that is serial dilution. We take the original sample, which is way too concentrated, and we mix it with water or something to dilute it 10 times. And then we take that mixture, we mix it with water or something to dilute that 10 times. So this is ultimately 100-fold dilution of that. Do it again, and we get 1,000-fold dilution of that. 10,000-fold, 100,000-fold, million-fold, 10 million-fold dilution. So we've diluted this 10 million times just by doing 1, 2, 3, 4, 5, 6, 7 steps. And 10 million is 10 to the 7th. So 7 steps makes perfect sense here. So that's all I'm going to say about pure culture. Watching me draw is boring and everything, but this is a fundamental topic in microbiology. We have to do this a lot. So we'll probably come back to this later.